Well, good morning, everyone. My name is Rebecca Wadham. I'm at the University of Virginia, and I want to welcome you to the third in our Cross BRC Tick webinar series. Today, we're going to be discussing the metagenomic analysis of ticks and how you could recreate such a study in the bacterial and viral BRC, which we call BV-BRC, which is a recent combination of the bacterial BRC, known as Patrick, and the viral BRCs, which are IRD and Viper. And let me point out that I'm ready for this webinar with my tick coffee cup. So let's see if I can go down. So this is, like I said, the third in our webinar series, but there's more on the horizon. And I encourage you to register for these at our sister BRC, which is ViewPathDB. The URL is at the bottom because there's a lot more to go. So today, I'm going to go over an interesting tick metagenomic example that I found in PLOS Neglected Tropical Diseases. It's a paper from 2019 by Ravi as the lead author, and it's a joint examination by Al Qahoud University in Palestine and the Quadram Institute in the UK. The scientists collected 200 ticks from dogs, sheep, and camels along the West Bank of Palestine, and they present a two-stage analysis to identify the ticks, the hosts, and the bacteria within. And they also went deep in detail to look for pathogens or what they think of as endosymbionts. So they have, I liked it that this is from their article. It shows the experimental design. They collected the ticks, extracted the DNA, and then narrowed it down to 14 individual samples. And on those samples, they did shotgun metagenomic sequencing, and then they did taxonomic profiling using the program Centrifuge. Now, at this point, they took the data and mapped it to mitochondrial sequences to identify both the tick and the host. That is something we couldn't do in BVBRC, but you could certainly map to the tick in our sister BRC, ViewPathDB, sorry about that. So that shows you that there's some potential to have some cross BRC analysis going on. Then they mapped the metagenomic reads to microbial genomes, something we can do in BVBRC. They assembled those reads. They took the draft sequences and compared them. They tried to pull out draft genome sequences from that, and then they did the comparison of those sequences. One thing I also like about the study is the data is available. That's one of the things that we need to be able to show you what you can do in BVBRC. So they have a bio project that consists of 14 experiments at the Sequence Read Archive, which I'll refer to as SRA from here on out. And I was able to look at all that data. Unfortunately, the 16S data is not available, so we couldn't look at that. They found bacteria in each of the samples. So here were the ticks that they looked at, and then they identified the ticks by morphology. They also identified the ticks by the closest hits to their mitochondrial DNA sequences. So sometimes there were differences between the ticks once they did that. And then in some instances, they were able to, doing something similar, identify who those ticks had been feeding on. They did taxonomic profiling, they used centrifuge, and then they were able to pull out genomes from the metagenomes for some of these species. They created phylogenetic trees. Now, their phylogenetic trees were based on SNPs. They aligned signature genes. I wasn't sure which those genes were because the article didn't go into that much detail or how many SNPs there were. And they built trees with RAXML. And then they did gene comparisons, not just of Rickettsia strains, but of everything that they found. And this was a little bit, they used something called Rory. And so this is a little hard to digest, but for those of you who are familiar with it, you'll get it faster than I did. GC content, and then the next ring in is GC skewed, showing the forward and reverse strand. And then each of these with colors are different strains that they were comparing to the reference strain, which was this MTU5 strain. Each of these, the darker color indicates 100% identity across the genes. The lighter color indicates less. So it would go from dark pink to light pink, light blue to even lighter blue, and dark blue to lighter blue for indications of homology. So if you've taken my other webinar in this series, and I'll be doing 2 more too, in each of these I explore what could I replicate in BV-BRC. These researchers went to a lot of different sources. They used a lot of different software to try to do this analysis. And remember, every time you're changing from one software pipeline to another, there's often some massaging of the data that you need to do. So think about that when you're doing your analysis. But I wanted to see how much we could do in our analysis. So for this presentation, I had so much fun with this data set, and I chewed on it and I manipulated it. And now I have a much better understanding of the data massaging that would make this thing better. Unfortunately, if I was going to do that today, to go to all the steps that I did, it would take too long. So I'm going to do a streamlined presentation. But I prepared data for you so that at your leisure, you can go in and look at things and make the actual comparisons. Because when I actually did the analysis, I looked at read quality first, which they also did. And then I did trimming and the taxonomic classification. And after the taxonomic classification, when I could see who were there, I aligned reads to specific bacterial genomes for mapping. And then I did metagenomic binning. I also did a comprehensive genome analysis where I could play with the different assemblers. And then I built trees and proteome comparison. But for today's presentation, we'll start with taxonomic classification. I'll go into our FASTQ utility service, which shows you how you can look at read quality, trimming, and alignment, even compare them all together. Then we'll do metagenomic binning, tree building, and proteome comparison. So let's get started on that. Oops. We've got to wait for, I don't think you guys can see my screen, so I'm going to have to stop share and then start sharing again of the screen and then this. So hopefully, can you all see my screen, which is the BVBRC here? Yes?
Yes.
Yes, looks good.
Okay.
Yes.
So this is the home screen for the Bacterial and Viral Bioinformatics Resource Center. Now we have tons of information about this. We've given tutorials, webinars, instructional videos. I can't go through everything today, but I want you to know that this is all here under the help tab for you. But let's say that you're a researcher and you know that the stars of their bacterial analyses were 3 different genera. Coxiella, they found a lot of Coxiella, they found a lot of Rickettsia, and they found a lot of anaplasma. So the first question would be, does your resource even have any of these genomes? Well, the first place to look is to go up into the organisms tab, and I'll make this a little bit bigger. And you can see we have bacterial pathogens, viral families, featured viruses, and then the cornucopia of all the data. And under the bacterial pathogens, you can see first, well, here's Coxiella. They're in alphabetical order. So I could click on that. And that will take me to the landing page for this taxon, which is Coxiella. Now, we've given webinars on telling you about each of these different tabs and how these pages are structured. So I don't want to go through that much information, but you can see that at least I can see 143 genomes. And if I click on the genomes tab, it's going to load those genomes so we can see how that's structured. If I click on filters here, you can see that under public, there are true genomes and false genomes. What does that mean? True means it's data that anyone can see when they log into the resource. So those are all the genomes that are public. And where do we get this data? From GenBank generally, although some people make data public here, or we get some from sequencing centers, or in some cases we go into the Sequence Read archive and pull large studies, specifically of antimicrobial resistance data for our machine learning classifiers. Now the false data, it's just private data, and this is only available to me. And you can see how much I've been playing with this data set because I've generated a lot of genomes from it. I just bring this up because I want to show you about the virtual integration we have at BV-BRC, the public data and my private data. Only I can see this. You're seeing this, of course, because you're watching my screen, but when you're logged in, you won't see this data. But I can draw from each of these sources to compare my data to any of the data within the resource. And you can see how seamlessly it's integrated, and that it's all been treated the same way. We're not doing apples to oranges, we're doing apples to apples. Everything is annotated exactly the same way. So the next organism was Rickettsia. So I could click on that from the organisms tab. You can see genomes here. And the third one of interest was anaplasma. And when I click on organisms, oh no, I don't see it here. Now, this is something that people say to me, not every week, but maybe every couple of weeks. I can't use your resource because you don't have my data for my bacterium. Actually, we have all the same genomes that are available in GitHub. These are just for rapid access. So how do I find that data? How do I find if you even have any anaplasma data? Well, you could go up here and use the global search that's on the top of every page. But if you were on the home page, you could find it here. And I could type in anaplasma, and that will take me, and hit return, of course, and that will take me to the—
Rebecca, can I interrupt with a question from a user, from an attendee? So their question is, did they, when you were looking at the Coxiella genomes, they thought they noticed that 20 of the 21 were poor quality?
Yes.
What does that mean? Maybe you can, or you can expand on that.
Well, we also run, we run quality scores on that, looking for completeness, contamination, too many contigs, things like that. If it was for my false data, my private data, a lot of those genomes were poor quality. And part of this lecture is about massaging that data to make it better quality, to be able to get a better assembly for that. So that's actually a pretty— let's go into anaplasma first. So I'll click on the taxa here from the, and let's see if we can find any of this. So here's the anaplasma, and I click on the genomes. And let's load up. And so then under quality, there are good genomes and there are poor genomes. So the poor genomes generally are tagging our flagging system, like too many contigs, too many hypothetical genes, signs of contamination. Those are the kind of things. But we didn't used to have this, and it helps you quickly. One of the things I do when I'm comparing my data to other data is I generally click on good. So I'm sure I'm doing good stuff and not poor stuff. Some of the stuff that's coming in from GenBank, some of it isn't necessarily good quality. So we want you to be aware of that. So I hope that answers that question. So I hope that now you have a sense of how you can find data that's not within the organisms tab here. But now let's talk about analysis of the data. So we go into tools and services, and today, like I told you, I first started off with read quality, which would be in FASTQ utilities under the utilities tag. But you notice we have genomics tools, protein tools, metagenomics tools. So let's start with taxonomic classification and click on that. And this is the landing page. I don't know where that red streak came from, but I don't know if you can see it, but I can see it. So I'm going to shrink this up. Here, shrink this up so you can see that the submit button at the bottom of the page is grayed out. It has to turn blue before you can submit this job. You could start with reads, or you could start with assembled contigs. But we're going to start with read files. Now, if you have your a paired read library, you can look at that. If you have a single read library, you could look at that. Or you could take something directly from the SRA, the Sequence Read Archive, and get that number. So I've got one from this study that I'll just put in here. And then once I have that accession number, I have to move it over into this selected read library box. And now it's there. And if I mouse over it, it'll tell me some information about that. Now, what's awesome about this is we didn't used to have this, and I would have to go into the Sequence Read Archive, download the read files to my computer, and generally they're big, so it would take a long time. So I get it downloaded to my computer, and then I have to upload it again to BV-BRC. This way, I can just put in the number. The developers said, you don't need to do that anymore. We'll just put in the accession number, and then you can examine it directly here. The algorithm we use currently is Kraken. They used centrifuge, and you can save classified sequences, you can save unclassified sequences, but you do need to establish an output folder.
Another question when you get a chance. Yeah,
sure.
Sorry to interrupt. So the question is, so in terms of the number of sequences, so this Claudia is asking, what if I have 192 paired read libraries? How do I upload those?
We just had a discussion about this morning because we have a user who has a similar circumstance, and we've been running a script independently for him on occasion to combine those files to make it easier for him. Or you could use command line to do it. This is the user who's having this problem that cannot use command line. So one of the developers has been doing this independently for him, and he's asked us for this so many times that this morning I was just telling the developer we need to create a service for this in the FASTQ utilities so that they can do this automatically without having me pull one of the developers off their task to do this. It's pretty easy for them to do it. But so I would say if you do have that, if you can work through command line, because in the resource you can find out about the command line tutorial and do things command line. If you're not good with that, just contact us through the help button, and until we get this service enabled, we can help you with that.
Okay, so the answer basically is that there is a way to do it using the command line, but using the web interface, that's underdeveloped.
You can do it 192 times, which would be
no, absolutely, but in an easy way. But and so the easy way to make it easy in the web interface, that's under development basically.
Right.
Hopefully will come at some point. But for now, there is a command line option if need be.
It's being asked more and more frequently, so it's become such a problem now of me having to ask the developer to do it independently that we were going to mention it, that we needed to do this right away, because I'm tired of pulling people off task to do it. So hopefully it'll come sooner. So you need a folder to put your data into. If you've recently created one, you can click on the drop down button to see it, or if you knew what it was named, you could start typing and it would show it, or you could do the folder icon, which will open up to your workspace where you could upload data or create a folder. But actually, I like Nobel Prize winning research because I want one, but I'm not likely to get it. So I need to name it, and I'll call this. It's good to name things really well because as you can see, these are the jobs that I've done, and over 3,000. And I often just like to give things very detailed names. And I know it looks stupid, but for me, it just works best. So you notice the submit button is now blue. That means I can launch this job. So I click submit, and it tells me the job has been submitted. And now it says it's been submitted successfully. If something went wrong here, you would need to let us know about it. So it says, click your jobs list. Well, how do I do that? You could go up here into workspaces and click on my jobs, or I always just go down here to the jobs list and click on that. And it tells me that this job is running. Now, how long is this going to take? Well, there are a number of factors that determine how long your job is going to take. One is the size of your reads. The bigger they are, the longer it's going to take. Another thing is how heavily used the service is, because we have sometimes as many as 30,000 jobs submitted a month by our users, not by us. We filter ourselves out. So some of our jobs are being heavily used, and so we have a queue that you just have to wait your turn. Sorry about that, but that's the way it goes. So are you gonna have to wait for this? No, we are not, because like a cooking show, I have run all of these things before, so we can look at them, and I've made them available for you as well. So let's click on public workspace, and then we scroll down until we see BV-BRC workshop. Click on that. And then we scroll down to tick webinars, click on that. And then for the metagenomic webinar, we click on that. I know it's a lot of clicking, and I'm going to be doing it a lot, so gird yourself for that. The manuscript I've made available here for you guys, so you could look at it here. But we were looking at taxonomic classification. So I'm going to click on that. And so that's going to show me the jobs that I ran under this. And one was just the analysis with just the reads, and then with the reads once I had trimmed them. So this checkered flag indicates, like NASCAR or Formula One, a successful race. So I can click on that and it will open the job. Now, in any of the tutorials, it's going to describe exactly what all of these things mean. Oh, look at how fast this one ran. Once it was able to run, 11 minutes was the total time it took. But, you know, if the queue's long, your job might wait for a day before it got to run. So to see something, which I want to show you this report, you click on the HTML, and then you click on the view icon. And this will show you a table that shows you the top hits. So you can see I have the percentage of reads to Staphylococcus is 4.16, to Coxiella is 2.84, and then down to Homo sapiens is 6.61. So obviously there was some human data on that. It also includes an interactive chart, which is kind of fun. So you click on that. And it's showing you that there were 62% that did not have hits. Now, we know this is from a squash tick, right? These 62% of the hits were to tick sequences. If we had some way to, well, we should think of some way that we can interact more with UPath-DB so that we could show some homology here. But you could see here's the staph and here's the Coxiella. And what's fun about this is you can click on this and expand it out even further. But I just wanted to show you what was happening. Remember, we've got the Staphylococcus here. and why I massage this data, and which I would encourage you to do as well. So this is a breadcrumb up on top that shows me my workspace. I'm going to go back to taxonomic classification, and I trimmed the same reads, and I'll show you why in the next part of this. So I click on the reads, and then I click on this taxonomic report HTML, and then I click on the view icon, and let's look down here. There's the Homo sapiens 6.73. There's the Coxiella 2.90. But what's gone? The Staphylococcus. So when I trimmed the reads, that it was showing that there was some interaction. The Kraken 2 works on K-mer identification. So whatever was in their reads that was trimmed away was interacting with Staphylococcus. So the Staphylococcus wasn't a true result. See, that's why it's important for you to massage your data, to analyze it in all different ways, to try to figure out what's going to give you the best hit. You could click on the interactive chart, and this is by Kronos. And there's the eukaryotes, and I click on bacteria to expand it. And now look, I'm seeing this Rickettsia massillae MTU5, and you can see the Coxiella here is 68%. If I go back to the job, this, remember that table is just a summary. I can look at the full report, and oh, is this painful to look at because it's huge. But I always do a find, and like I can look for the Coxiella pretty quickly and see that 2.9% of the reads map to that. That doesn't seem like too much to me. And then if I looked at the Rickettsia massillae, only 0.3% or 0.21% did to this MTU5. So the question we're going to be asking in the rest of this webinar is, how many reads does it take for you to even be able to pull a genome out of this? So this is the taxonomic, this is the end of the taxonomic classification stuff. But what I did originally, and the reason I bring this up, is it gives me things I can align my reads to try to game the system and be able to get better results. Well, one of the things you need to do to massage your reads is you can go into FASTQ utilities. So I click on that, and I would need to put it in the same folder, which is my Nobel Prize winning research. And it would be, this is QC line 2 Rickettsia. I'll just call it that. So I need to give it an output name. So you already know what I'm going to do. I go into the pipeline, and I can do these things individually, which I did when I ran this the first time, which I looked at FastQC, which is for read quality, which our users are, sorry, the authors did as well. And then I trimmed it. And then I can align. Now watch the target genome here. I can also align the reads after they're trimmed. So you can do these all individually, or you can do them one at a time, but I have to do a target genome for this. So this was, it was called MTU5, was that target genome. And see how simple this is? Something like that, just putting in the strain name that I could see from the taxonomic classification report, I can find that genome. And now what this service is going to do is it's going to look at read quality, it's going to trim the reads, and it's going to align them to this genome. So if I had paired reads or single reads, I could run those, but I've still got my same SRA accession. Whoops, and I've got to move it over into my selected library box. And when I do that, the submit button turns blue, and I can submit that job. And it tells me it's being submitted, and then it'll tell me it has been successfully submitted. But like I said, this is like a cooking show. I already have those jobs available for you. So let's go into public workspaces, under workspace, scroll down to BV-BRC workshop. scroll down to tick webinars. And this was the first webinar I gave. This is the second one. And now we're in FASTQ utilities. So let's click on that. Now this is the job where I did FASTQ utilities with just the reads only. So just to look at read quality. So I'm going to click on this one. And here it's showing me that job with the checkered flag, meaning I was successful. So I click on that and it opens it up. And I just want to show you what this report looks like, the FASTQC quality report. So I click on the view icon and it tells me that none of the sequences were, it tells me information about that. And you can see here that there is, if you scroll down, that this last read pair, the last ones are dip into poorer quality. I'm just going to scroll through these things because none of this was too remarkable to me. But what I wanted to point out was at the bottom, the adapters, it's an indication that there's a high number of adapters in that. So let's go back to this folder and then see what happens. I want to show you what the report looks like when you trim these things. So I clicked on this and then the checkered flag, and this is a trimming report. So I highlight that and then click the view icon. And there's a lot of information here, but it tells me how many of my reads had adapters, how many were quality trimmed. And then let's go back up and let's look at the report after that. But before I click on that, notice that here are my reads that have been trimmed. Now these are in my workspace. And if I want to use these for analysis, instead of going to the SRA or entering that accession number, here's what I can use once I decide that their quality is better. So here's the at the quality report after they've been trimmed. So I click on that and look at this. It's at the very end, the data is better at the end of those reads. And if we go up, sorry about the scrolling, no, we go all the way, look, the adapters are gone. Now, why does this matter? When we looked at taxonomic classification before and after trimming, before trimming, there was Staphylococcus. After trimming, there wasn't any. Those adapters that were trimmed away were what was hitting the Staphylococcus. And I only bring this up to tell you, to make you think about all the different ways you need to look at your data to ensure its quality, especially before you move it to publication so that you don't make any mistakes. And then the last thing we did was align the reads. And I just want to show you the alignment files. So I aligned them to the Coxiella genomes that were suggested before and after trimming. And I did that with the Rickettsia too. The Rickettsia is a better story, so I'm going to show you that. This is before trimming. So I click on the checkered flag. And there are a number of files that come with this, but including this are the read files now that mapped to that genome. So if you like this, you can take it into assembly, you can take it into metagenomic binning, you can even compare it to a reference genome if you want. But let's look at the report, which is the SAMstat HTML. That's the report on the alignment. And the main reason I like to look at this report is for this dumb circle, which makes me think of Pac-Man. And what it's showing me is that a lot of those reads are unmapped. When I try, a lot of those reads were unmapped in the original sample. When I tried to map it to the genome that taxonomic classification told me was in my sample. But only 0.4% of those reads mapped there.
So there's a question
Yes.
from Tomaskin Samuel. He's asking, how sure can you be that staff didn't belong there?
Oh, because I tried to map the reads to it. It didn't map to anything. So I tried to drag everything out from it. So that's why I'm convinced it wasn't there. Because I tried mapping to staff itself and I couldn't find them. So
Okay, good.
That's how I feel convinced of it. Of course, other people would do it different ways, and probably I'm really just a biologist. I don't have any coding skills. And so this is the way I would do it. I'm sure a straight up bioinformatician would twist that data in a lot of different ways than I did, but I feel pretty confident that that was a mistake, that it was a misidentification. Remember that taxonomic classification is just on K-mer signatures, which is small pieces of DNA that are unique to certain genomes. So, and it wouldn't be outside the realm that an adapter sequence could align to a Staphylococcus genome. Okay, so these are from the straight up reads. And let's go back to look at the alignment just because this one is fun. So that was 0.4%. After I trimmed them and I looked at it, and once again, here they are trimmed and aligned. So it lets you know that. Let's look at that and click the view icon. And there's the Pacman thing coming up. Now it's up to 0.9%. And this tells you the number of reads that are high quality mapping to that. And I first looked at that and I thought, well, that doesn't seem like very much, and I doubt I can get a good genome out of that. But let's see what we can do. So we've done taxonomic classification. We've looked at the reads, and we've looked at the quality before and after trimming, and we've also done alignment. Now at this point, you could go into tools and services and do assembly or even a comprehensive genome analysis on those reads, where I could put that in so I can find them, and I could do the aligned ones, or I could just do the trimmed ones. You can see this is the trimmed and it tells me who it is. And then with the aligned ones, it tells me who it was aligned to. And the reason I might do that is so that I could do unicycler or spades. I'm not going to go into too much detail for that today, but unicycler did give me a better assembly in that it gave me fewer contigs. But what I really want to show you is how you can do the metagenomic binning service that we have. So you can start with reads or assembled contigs, but we have the reads. And at this point, I could enter those files, or I could do just from a straight up SRA sample. Look at that. And what this service does, and you can find out more about it in the tutorial, and it has been published as well, is it tries to draw out genomes from a complex mixture. And so I can choose whichever assembly strategy I like. I actually prefer megahit to metaspades. I don't know, it just gives me better results. In this example, I would encourage you, you know what? I do everything when I do these things, especially when it's my data and I'm moving toward publication. I want the very best assembly. So I'm trying everything I can to get the best assembly, to get the best results. So I would encourage you to do that too. And believe me, in this, I've done everything with this thing. And I just want bacteria. I want to put it in my folder. And I've got to give it an output name, MB. And you can tell I've run these jobs a number of times on each of the different samples. Remember, there were 14. So I've done a lot on this. And actually, I have to put in a genome group name too. And then I can submit that job. So that job's been submitted. So, but like everything else that I've told you, I've already prepared this. So let's go into public workspaces. And let's scroll down to BV-BRC workshop. and the tick webinars, and then the metagenomic webinar. I know, lots of clicking. And then this one is metagenomic binning. And so I have a straight up binning report, and then the reads that were mapped to Coxiella, I've submitted those to metagenomic binning. And then the reads that were mapped, trimmed and mapped to Rickettsia. So first, I'm going to show you these two. Let's look at what it looked like, what the metagenomic binning report looks like when we just mapped it straight to the SRA reads. So I click on that. And once again, you can go into the tutorials to find out what all of these things mean, but I want to look at the binning report. So let's click on that. And it tells me I was able to get a good bin and a bad bin. What's a bin? A bin's a genome. So the good bin, the good genome, was what is identified as the Coxiella endosymbiont, and they call it the clonal population. And they found it was 100% complete with 0% contamination, but it had a few problematic roles, which we'll go into in a minute. And then it found the bad bin was to this Rickettsia macellate clonal population. Now think about this. When this particular sample, only 0.4% of the total reads, and I can't remember exactly what the figure was, but that small percent mapped to this genome. But it was able to pull out, the 94.5% complete genome out of this. But you notice this yellow color, that's indicating that there were flags raised on this particular genome. It said it had 15.6% contamination, and that almost 90% of the genes annotated on it were hypothetical. So that's a red, I would say a red flag, but it's a yellow flag, indicating that there are some quality issues. And remember that in the tutorials, it describes what each of these different column heads mean. So I want to go back and look at what happened after we did the trimming and mapping to that Rickettsia MTU5 genome. So I click on that, and then I want to see that binning report and view that. And suddenly it says it's a good genome now because the contamination has gone down and the number of hypothetical genes have gone down. And this is once again to emphasize to you how important it is to massage your data and to try to do different things to it to get the best quality data you can to increase your chance of publication. Now, this genome, if I click here, it's going to show me the genome that it's now nestled into the BV-BRC website. It looks just like any other genome there. All the data is there. And if you were to click here to give you an example of what these problematic roles are and what that even means, these are genes. Then here's another flag saying the N50 for the contigs. It thought for this one was pretty high. It also, so let's talk about what this means though. With the problematic roles, it means genes that they had problems with. So let's start with this gene. So when it looked at this gene, it says, most of the Rickettsia, the reference genome we compared it to, it only had one. Your genome had two, and these were two different genes. And when we looked at it, this gene, this is the genome ID here, which you can see up here. It's the unique identifier for this genome within the BVBRC. And this says, this is protein encoding gene number 124. This was the 124th gene that we called that it was in it was the part of this gene. And it says here it ends at the end of this contig. And then there's another gene here, the 577th gene, that ends at the end of this contig, which is short. So what does that tell you? That shows you how you can even use this further. To me, it indicates that these two contigs could be joined up. and that this genome actually has one gene for this, but it's separated on 2 contigs. So I just wanted to show you how you can use this kind of report to improve your data if you wanted to go in and stitch those 2 contigs together, which the assembler, in this case, Mega Hit, was not able to do. So pretty cool, right? From a complex sample, bringing out these genomes. And not only that, if I trimmed and aligned the data, I was able to improve the quality of my genome. So we've done read quality, we've done taxonomic classification, we've done metagenomic binning. The next thing they did was building a tree. So we go into tools and services, and under genomics, click on phylogenetic tree. So I want to go back and bring that over. Can you guys see that tree? I hope.
Yes.
Okay. So this is from their publication. And one thing that they did that was very useful was they provided the assembly accession. What wasn't useful is that in some cases they got the letters wrong. Some of these are GCA, not GCF. And this actual accession is to a closely related but different genome. But nonetheless, you know, everybody makes mistakes. It's not a big deal. But I just want to show you what you can do with this kind of information. I could search for these things by name, but once I have the assembly accessions, I can grab those and I can go into the global search tool and enter them there. And then I can say, I want to find genomes and I want to find any exact term. So this is the way we parse the data. Now this was working earlier, so it's being problematic now, but you should be, oh, it's the wrong, sorry, it's the ones with the GCFs and not the GCAs. It was the former way that they originally had the data. So let me go over here to BV-BRC. Come on, guy. And these are the corrected ones. Sorry, it took me a while to get this. There we go. And then the genomes and the any exact term, and click on the search button. And then here the genomes pop up. And if where's my little guy again? And if I bring those over, I've got my genomes here. And then once I have this, I can click on this and I can create a genome group. Now it's not so hard when there are just a short number of genomes. But when you have like 100 genomes, you don't want to enter them individually into the service. It's like the person who had 192 paired read files that they want to enter for an assembly. Nobody wants to do all that clicking. So if I can do it easily here and add them to a new group, I'll call it Ravi Genomes 19 April 2022, and add that, and you can go in and clean things up later if you want to delete groups. So I add that to a group, and the main reason I'm showing you that is I can go into tools and services, click on phylogenetic tree, and then it should show me the group I just created. So I can add that, but I want my private data to be added as well. I want to, because the whole point of this is to say, my quality, the genome I extracted from this guy, I want to see that too. I want to see where it is in this tree. You have to be able to spell. Oh, I was so busy talking. When I click on this, it shows me that I can filter things based on public genomes, which are reference, representative, or all public genomes. And then I can just say, I want to see my private genome. So I've got my private in there. And then I start typing, and BVBRC is going to try to help me. And it says, I'm going to help you find all the genomes that match that. This one is my actual favorite. So I'm clicking on that and adding that. And then I go into the output folder. I will like to put everything into the same data. I'm going to call it Ravi genomes. Clonal, oops, I could spell, 19 April. And then I want a powerful tree. Now let me describe what this service does. This is our codon trees pipeline, and it takes perfect protein families, any genome that's annotated in and all the public genomes and your private genomes, when it runs through the annotation service, they're all annotated using the RAS-TK pipeline. And part of this is assigning protein families. This service, which is called the codon tree pipeline, looks for what we call perfect protein families. And in this instance, it means a protein family that has one gene for each genome selected. So in each protein family, if there are 6 genomes here, it would have 6 representatives, 1 from each genome. Those are the perfect families. Then it takes the nucleotide sequence and the amino acid sequence for both of those. So the nucleotide would be more like a SNP pipeline, but it's also adding in the amino acid sequence for each of those 500 genes. It concatenates them together and builds an alignment, and then uses RAXML for the tree. 500 is a pretty powerful tree, so that's what I want. How long will this take? If you have more genomes, it's going to take longer. If you ask it for 500 genes, it's going to take a while. If you ask it for 1,000 genes, it can take a really long time. So be careful what you wish for, but I'll submit that job. And so it submitted it, and then it says it's been queued. So let's go see what it looks like from the job I ran earlier. So I click on workspaces, public workspace, scroll down to BV-BRC workshop, TIC webinar, metagenomic webinar, and now we're in phylogenetic tree. And so
Rebecca,
yep.
Rebecca, quickly, I think I missed this chat a couple of minutes ago, but Claudia was wondering whether the data was from a 16S or from a whole.
It's whole
genome,
genomes.
shotgun.
Whole genomes. Okay. It's from whole genomes that have been. They have to have been assembled and annotated within the resource. You can't bring in outside data, and it's a requirement that they have to be annotated here because they're drawing on our protein families.
Right. Right.
Okay. So this is the one I ran yesterday, just with those genomes. So let's click on this. And once again, you can refer to the tutorial to find out information about all of these files that come with this service. But let's look at the genomes report and click on the view icon. And I'm going to shrink this up a bit so you can see it. So here are the genomes that Robbie et al. Used. And here's my guy, the ones that I was able to draw from that accession number. Now,
there are a few more questions. Sorry to keep interrupting, but I just want to make sure we don't go too far.
Sure.
So there was a question from Kingsley asking about, I'm just going to read it off directly, but he was saying, I was expecting the accession numbers as part of the data pulled up to be sure they were the same data, especially when you select exact terms. So
right.
So why weren't they? So I guess the question is, any reason, I guess, for this?
Oh, why did I use different accession numbers?
Yeah.
Oh, because I realized after that, when I found this particular genome, it should have been GCA. He just made a mistake. They just made a mistake when they recorded this. 3 of the 1, 2, 3, 4, 4 of those, 5 of those actually were GCA, and they put GCF. And this particular accession number did not map to strain AT24. It mapped to strain Tate's Hell. So
there are mistakes in the publications, and
there were some mistakes in the publication.
So Rebecca, I can add to that. So actually the GCF accession number correspond to the RAPSIC assembly, and GCA correspond to the GenBank assembly. Even though the assembly is exactly the same, they kind of correspond to 2 different databases, but they are pretty much the same.
Well, that's even more information. So they didn't make a mistake. It's just they used 2 different sources, but I was able to find the data pretty easily. What's the next
So
question, Omar?
Yeah, hopefully that answered your question, Kingsley. If not, just type in another question, or feel free to unmute yourself as well. So there are a couple of questions also from Claudia. I think she sent them as a direct message instead of to the general. So one was, how do I know which genomes I should select? I guess that's a more general question when you're selecting a genomes.
Okay. I would first select genomes that are good quality. Another thing I would select is, if I was making a tree, I would look at reference or representative genomes. These are the GenBank gold standard genomes that they have established. So they're generally good quality data to look at. And then you can just branch out. And, I build tons of trees, and I go in and I do my own pruning as well, because when I'm going to publish, I want it to be the best tree possible, and I want to have the best representatives. So I am very careful on what I include. And I know it doesn't sound vain, but I also limit how many genomes I will include in it, because I want it to look good in a publication. So at first I'll run everything, and then I'll start pruning down. But also this report, and I'll show you this will give you some hints about what to include. Okay. So first of all, go ahead. Is there another question?
There are more questions, but go ahead. Yeah.
Okay, let me finish with this one, and then I'll go back to the questions. So we've got this guy here. Whoops, now I've lost my little friend. This is our clonal one, and they have their clonal one next to Africa, which is, this is equally related really to all of these genomes. So which one is right? Well, I don't know how many genes they used or how many SNPs they used in their publication, but in mine, we say I asked for 500 genes, it found 500 genes, and it used 192,000 nucleotides and 192,000 amino acids and 578,000 nucleotides. But that still doesn't tell me which one, which orientation is correct. So I ran with the way I do that, and this I think will help answer, I believe it was Claudia's question. I go into phylogenetic tree, and here's another one I added with better genomes, or more genomes, not better. I included more genomes, and I click the view here, and here's the Africa, and here is the clonal population here. So amongst all of those. Now this will help with Claudia's question. If I asked for 500 genes, this one was only able to find 409 that met that initial criteria of perfect families. And so, pardon me while I scroll, because there's a lot of information associated with this report. It shows you all the genomes and all 409 protein families that it used. And remember, it's taking the nucleotides and amino acids from each of those and concatenating them together. But at the very bottom, strategies to increase single copy gene number. And it says, you asked for 500, you got 409. And it gives you genomes to omit to make it better. Often those are poor quality genomes that it's asking you to get rid of. So I often do a search and find for that to see where that guy is in the tree. And it's saying if I eliminated this guy, I would be able to add to the quality of that genome. Okay, let's take some more questions before I go to the next one.
Okay, so there are, I guess, two more general questions. So one is about using specifically 16S amplicon metagenomic sequencing. And the question is, this is again from Claudia, can she build a tree based on those? And can she get any functional predictions from the 16S? And I think, obviously the quick answer is that, well, it's if you're sequencing 16S, then you're limited to 16S.
Right.
Yeah.
You can build a gene tree with 16S data, but it would have to be annotated within the system. So I would have to go into those genomes, click on, well, I can show you how I would do it.
You could, while you're doing that, she could, obviously maybe not within BV-BRC, but she could take her 16S data and generate a tree using a separate tool, right? With some kind of an alignment.
We do have gene tools to be able to do that. So
okay, great.
Let's say from a genome group that I just created. And let's just say, I like this one better. So I want to see the genome view. And then I want to go to the proteins, although these are genes too. And then let me open up the filters. I want to click off CDS. I want to get my, see if I can get the 16S this way. I'm trying to get my, there we go. Okay, ribosomal RNA. No, I don't want CDS. I just want ribosomal RNA. And then we have different names for it than most people, but I'll just put in large. Click on that. Click here. Come on. And then I would create a feature group of that. Let's see if I could do it directly from here. Now, well, I could create an MSA from that, which also has a gene tree, but we also, if I created a feature group of that, which would be features, oops, sorry, I meant to create a group. So
ultimately, sorry to interrupt, but ultimately a user could also upload their own 16S data, right? And generate a tree. Okay, okay. No. So they would have to do the,
actually,
if they have their...
actually the gene tree service, let me see if it'll, I can't even talk. See. Oh yeah, well, yes, she could. Here, this is a new service that I haven't, this is one of the services that came integrated in with IRD and Viper. So I haven't been using it extensively, but yes, she could bring it in here. And using this tool, I could add my feature group. I could upload my 16S sequencing, and then I could create a gene tree from that. Make
okay,
sense?
that's it. Yeah, yeah. And there are many questions coming along. I don't know how much more you wanted to cover.
Let me do one last service really quickly, and then we can. Or
okay.
I don't know. What do you think? You
no, go ahead. Maybe Ron or Malik can go ahead and reply. Sorry, answer in the chat as well. I can answer as well, but they will be better equipped for those questions. So yes, go ahead.
Okay. So one of the things they did was this gene comparison. And can we do something similar? Remember, these were 3 different genomes, and the colors within each ring, stronger colors meant less homology, lighter colors meant more, or stronger colors meant more homology, lighter colors meant less homology. And this was a tool by Rory. Well, we have something similar, which is our oldest tool basically in the resource, which is the proteome comparison tool. So I could put in, oops, Nobel Prize winning research, because that's where this is going. And it is the, and whoops, it helps if you can spell, Rickettsia comparison, Robbie. And so let's look at the genomes they used and see. I'm just trying to show you different ways that you can look at this. Now, what this service does is bidirectional blast hits. So they're looking, you can compare up to 10 genomes. So this is a very fine-grained response. And this is the reference genome. And they used Rickettsia maculae MTU5. And so even just clicking on that, see how easy that is? And I think it was Claudia. This is a when it says rep here, it means it's one of GenBank's representative genomes, which is generally a high quality, well-trusted genome. So I click on that. That's my reference genome, the same reference genome that Robbie et al. Used. And then I want to add in, you could do it as a group. Here you could add in FASTA file, feature group, genome group, but I wanted to select the genomes. They did this Rickettsia, Rickettsia, if I've spelled it correctly. Oh, look, so, and it was strain ECT. So I've got that, and I can add that in. And then they did Macile AZT 80. And I'm just trying to show you how you can take different things here. Now look at this. There are two of them here. Which one do I use? I think I'll use this one. This indicates to me, I think it's a second initial sequence by, I believe it's Rocky Mountain Biological Lab, but I'll just choose this one. And then I had my private genome that I wanted to look at, which is, in this case, I could even add the genome ID number for my genome that I liked the best. And that's my private genome, and add that. And now the submit button is blue. So I can submit that job. But once again, I've baked my cake, and it's all here in the public workspace. And go down to BV-BRC workshop, tick webinar, metagenomic webinar, proteome comparison. And here's the single job that I ran. Clicking on that, and once again, refer to the tutorial for all of these things. But this time I want you to click up here to the view icon. And this will show you this job, which shows you from the outside to the inside of these bidirectional blast hits. This is the MTU5 genome. This second one in is the ECT genome. This third one in is the AZT80 genome. And there's my clonal population, my private genome. And this is showing you the strength of the blast hits between each of the proteins annotated in these genomes. And it also is, the color is also an indication of bidirectional or unidirectional hits. So if it's dark blue or purple, that means a really strong hit. If it gets into the yellows or reds, that means a less strong hit. So I'm going to pull up this and show you the comparison between the two. So this one, at least we're using the same colors across, so you can see things, I think, a little bit differently. Another thing I want to point out that comes with this tool, that's probably the most awesome thing about it. I often see this particular figure in publications, because I look at all the publications that cite our resource. So I provide this, but you could go into download and you could click on the genome comparison table, either in text or in Excel. But I've already got that for you. And I don't want to go into all the details on this thing, but this is what it looks like. Hopefully you all can see this. This is my reference genome, and I'd encourage you to look at the tutorial to see what each of these column heads mean. But then you have each of the comparisons. Now notice that the colors are matching the colors here, like these red things here are the red ones here. Not only is it telling you bidirectional or unidirectional, it's telling you the direction of the unidirectional hit within this genome. It's telling you the things it's missing, which you could see in the diagram. And these are my two favorite columns here, which is showing you for this particular gene, the percent identity and the sequence coverage of that. So it's so awesome. It's such a great tool to be able to scroll through a genome. I use this all the time to look for areas of lateral transfer and things like that. It's just very powerful. So they did, we did read massage. We did read massage was quality, trimming, aligning. We did taxonomic classification. We did metagenomic binning. I also talked briefly about running genomes through the comprehensive genome analysis service where I could play with the different assembler. We built trees. We did proteome comparison. So the last thing is let's compare the results. But if there are questions, I can pause here and we can do that in a second. Are there questions that I need to address, or do you think?
I think everything has been covered. There was a question about constructing phylogenetic trees using viral genomes, but I think that was answered. Yeah, I see Malik answered that.
Okay.
So I think everything was answered.
Okay, so let's see how we did. So are you guys able to see the slideshow?
Yes.
Okay, so but for the bioinformatic tools for read quality, Ravi et al. Used FastQC, as did we. For taxonomic classification, they use centrifuge, we use Kraken 2. For trimming, they use Trim-O-Matic, we use Trim Galore. Read alignment, Bowtie 2, our process also does Bowtie 2. They did their own metagenomic binning using Bowtie 2 and something I'm not familiar with, which is QualiMap 2. And we have our metagenomic binning service. For the annotation part, for the assembly, they use Spades. I used MegaHit, Spades, and Unicycler. Actually, I thought Unicycler gave me the best assembly on that. For annotation, they used Proca. We used RASTK. For genome comparison, they used Rory. We used proteome comparison. And then for tree building, they did SNPs and RAXML, and we used nucleic acids and amino acids in RAXML. The one difference is all of the tools on the right are united within our resource, so it's a seamless way. You don't have to change any of the data between any of the different steps because it's all united seamlessly. So let's look a little bit at the results. And for taxonomic classification, remember this showed you the ticks, the tick morphology, sequence, host, and the key microbes in that, and also from the metagenome. But let's look at the microbes. Remember, they use centrifuge, we use Kraken 2. For each of these accession numbers, this is what they found within them, and this is what we found within them. And I went through and I also looked at the percent of the reads that mapped to each of these. So we always got what they got, except in this single instance for this SRR number that ended in 9.6, they were able to draw out Coxiella. We didn't see that. And in some places we were able to see things like an additional Rickettsia that they did not see there. So I think all in all, it worked out pretty well. And then for metagenomic binning, and remember we used two different services here, that with their process, which was using BLAST and that QualiMap 2. And these were the genomes that they could pull out of that, compared, and then here's what was pulled out with ours. Now this was, I kind of also, because we can rank the genomes as good or poor. So they were getting those genomes, we got these, and in some cases where they were not able to get genomes, we were able to get genomes. Some of them are poor quality, but some of them are good quality, and some weird stuff that I've never seen before, Nevskia, no idea what that is. So we were able to get some good genomes from here. Some of them are good, but we were able to see more from the metagenomic binning service that we use than they were able to see. And then for the phylogenetic trees, they did it, we did it, and then also for the proteome comparison, the two different tools. So I think I've been able to show that you could do a very similar analysis strain, especially from the microbiology part for the bacterial part, at the BV-BRC. And then you could also do the identification of the ticks at ViewPath-DB. But that sort of concludes what I have today. And if you have any more questions, I'm here to answer them for you.
That was great, Rebecca. I don't see any more questions. If anybody has questions, please, again, feel free to unmute yourself or just type in the chat. Otherwise, I know we went over the hour. So
Sorry.
feel free. No problem. No, this was great. I think people found it useful and lots of people stayed on. And also, obviously, this was recorded and will be made available. So if anybody had to leave or joined late, you will get a link to the recording and will be able to watch it at your own leisure. So maybe we'll hang on another 10 seconds and see people if people are asking questions. Otherwise,
it said that somebody was raising their hand, but I really can't.
Oh, I see. Dina,
yes,
I mean, did
yes,
you?
please.
Yes, go ahead, Dina.
Thank
Sure.
you very much for this useful seminar. I am a microbiologist, so I am interested in the bacteria in the ticks, but I couldn't deal with the eukaryotic genome.
Right.
So how can I separate and work only on the bacterial genome?
You would use that. You would go into tools and services. First, I would run taxonomic classification to see who was there. And then I would try to map those reads to the bacteria using the FastQC and then the align. Because remember that the taxonomic classification, and let me go back to that public workspace. Sorry, I know you all are sick of, you're probably going to hear it when you're in your sleep tonight. BV-BRC, tick webinars, metagenomic webinar. Okay, so when I'm in taxonomic classification and I'm looking at that job, the report gives me an indication of which genomes are in there. And this is showing me that this one is mapping to, that I can see this guy, this Coxiella mudroy. So I could align those reads to this and then extract, throw away everything else. So once I have done that, let me go back to the job. Let's see, I want to go back to this one. So that's the taxonomic classification. And if I then ran, I knew who it was mapping to, then I could use the FastQ utility service and go in and do an alignment. And then I could see those reads. Let me show you. Then I could use these reads that were aligning to that genome. And then I could use that in there. What would actually probably be more useful for you, but not for the ticks. Actually, that's the best way to do it because we don't have the tick genome, and that's the easiest way, I think, for you to do it. Does that answer your question?
Yes, thank you very much.
And remember that some of these, especially with the Rickettsia, there were very few reads there, right? Very few reads, only, 100,000 reads mapping to that genome. But after I aligned it to what the taxonomic classification identified for me as what I was showing strong homology to, I was able to get a good genome from that. So I may have been lukewarm about this service before. Now I am an ardent supporter of alignment followed by taxonomic classification.
Yes, thank you very much.
Sure.
There's another question about, we talked about loading FASTQ files, and that's in general what everybody works with. But the question is, can you load FAST5 files? I think those are the ones coming from nanopore sequencing.
Well, yeah, we do. Yes, yes, you can load. We do nanopore alignments. We would use, as I recall, aren't nanopore, when you do an assembly, I believe nanopore, they're long reads, are they? Come on.
Yes.
Yeah. So you would load those and you then use Canoe as the assembler. But you have to be sure that they're tagged as reads. Now, if I upload something here in this, when I'm within this, the assembly app, it's going to look specifically. Let's see if I have anything in my download file, in my upload file, on my computer.
And Robert answered in the chat that it's possible that we don't directly support FAST5, but he'll check on that.
Yeah, well, there's someone on who I know could, but see, when I'm looking here In my computer, it's looking specifically for things that would match, that has the right format in for that. If you upload it yourself into the workspace, let me cancel here. If I'm in the workspace or my home directory, come on, workspace, let's go. And I upload here, I have to be sure that I would specify those things as reads so that the service can see them.
There are also tools that allow you to convert between FAST5 and FASTQ, so that may be an option as well. Depending on what you're trying to do, yeah.
Yeah, we should also provide that in our FASTQ utility service. So for the developers who are listening, think about that as well, because that might be useful for people, especially since we've had some people with BAM files too. Anyhow, anything else?
No, I think those were all the questions. Any last minute thoughts? And of course, I think, as usual, I know Rebecca probably said this earlier, but if you have any questions later on, don't panic. This is not the only time you can ask questions. Just click on the contact us link on our websites. We have different ways. Yeah, you see where she's pointing, exactly. Whether you're using BV-BRC or ViewPathDB, there's a way to contact us, and we'd love to hear from you and answer your questions. So definitely send us emails. And of course, we have additional webinars, which we did provide the link in the chat, but it's the same page where you registered for this webinar. You will see additional webinars in this TIC series, and each of our BRCs also conducts separate webinars as well, which are also posted and highlighted on our different pages.
Oh, one other thing is BV-BRC, the bacterial part of it, will be at ASM Microbe in June. If you're going, I encourage you to go on June 9th and sign up for the workshop, because we'll be giving an all-day workshop there where we step through this stuff, and we'll be there to help you. And there'll be some, if you want face-to-face time, I don't know if anybody wants that anymore, but we'll be there. And I bring chocolates too. So.
And this will be only in person, I'm assuming. There won't be a virtual. It's not a hybrid.
No, it's only in person, and it's sponsored by ASM.
Yep. Great. I think this is it. It looks like everybody's happy. Lots of thank yous to you, Rebecca. Great job. And so thanks, everybody. And again, you will get an email with the recording, and it'll also be posted on the website. Thanks, everybody, and have a great rest of your day. Yeah, thank you.
Bye.
Bye-bye.